Hello everybody and welcome to my talk. My name is Amani Ballah, Assistant Professor of Pharmacology and Toxicology, Faculty of Pharmacy, Al-Azhar University. My talk is titled as Omeprazole activates the profibrotic gene's expression in rat kidney, implication of TGF-beta Smad and ERK1/2 signaling pathway. First, I would like to give you short notes about proton pump inhibitors. Proton pump inhibitors are a group of drugs with strong ability to block gastric acid secretion by inhibiting the action of proton pump that's found in parietal cell of the stomach. They are originally designed to help cure gastric ulcers. However, the use in combating acid reflux has led them to become one of the most popular drugs worldwide. Problems started being reported when this powerful drug were made available over the counter. Omeprazole is one of the most commonly prescribed proton pump inhibitors. Several studies have reported that the use of proton pump inhibitors may be associated with serious side effects, including gastric neoplasia, hip fracture, dementia, acute interstitial nephritis, acute kidney injury, also chronic liver and kidney diseases. It has been reported that TGF-beta Smad signaling activation play an important role in the expression of profibrotic genes, leading to tissue fibrosis. Usually, TGF-beta is secreted in latent form. Many factors activate TGF-beta. The activated form liberating and binding with TGF-beta receptors, resulting in Smad phosphorylation. This phosphorylated Smad activates the transcription of many target genes, including the profibrotic gene, connective tissue growth factor, and tissue inhibitors of metalloproteinases, leading to tissue fibrosis. Also, it has been reported that EGF receptor mediated ERK1/2 phosphorylation play an important role in the induction of cell proliferation. Adam-17 is implicated in the liberation of pro-HB EGF, resulting in the release of the active form of HB EGF, which activates EGF receptor, which in turn activates the ERK1/2 signaling cascade and activates the transcription of many target genes, including cyclin B1, and subsequent induction of cell proliferation. However, the use of proton pump inhibitors is associated with increased risk of acute and chronic kidney disease by mechanisms not entirely known. The present study was designed to investigate the molecular mechanisms by which omeprazole causes chronic kidney disease by examination of, first, the potential effect of omeprazole on TGF-beta Smad signaling pathway and subsequent profibrotic gene expression. Second, the potential effect of omeprazole on ERK1/2 signaling pathway and subsequent expression of cell proliferation genes. Experimental design. Experiment 1 was designed to investigate the effect of omeprazole on TGF-beta activation and subsequent Smad phosphorylation, as well as ERK1-2 phosphorylation at different time points. So we have 10 groups. 60 animals were divided into 10 groups, 6 animals per each group, control groups and omeprazole groups. Experiment 2. 72 animals were divided into 6 groups, 12 red per each group. Group 1 serves as control, Group 2 omeprazole group, Group 3 omeprazole plus TGF-beta inhibitor, Group 4 omeprazole plus EGF receptor inhibitor, Group 5 TGF-beta inhibitor alone, and finally Group 6 EGF receptor inhibitor alone. Half the animals, 6 animals per each group, were sacrificed 4 hours after drug administration to detect the activation of ADAM17, TGF-beta, as well as analysis of phosphosmad-2,3, phospho-ERK1-2, and phospho-EGF receptors. The rest of animals, 6 animals per each group, were sacrificed 24 hours after drug administration to detect CTGF, TIMP-1, and cyclin B1 expression, as well as SOD activity and MDA content. Coming to the results. In the first figure, we can see that the administration of omeprazole significantly induced TGF-beta activation, as indicated by an increase in the renal level of active TGF-beta, with peak measured after 4 hours, which thereafter declined to the level of control group after 24 hours. TGF-beta activation induced by omeprazole has been translated into Smad activation, as demonstrated in figure B. Smad phosphorylation was detected in kidney tissue from rats treated with omeprazole after 4 hours, which thereafter declined to the level of control group after 24 hours, indicating that omeprazole activates TGF-beta Smad signaling pathway in rat kidney. In this figure, to investigate the involvement of TGF-beta in Smad phosphorylation induced by omeprazole, rats were treated with either omeprazole and or TGF-beta inhibitor. And as we can see, concomitant administration of TGF-beta inhibitor along with omeprazole significantly reduced TGF-beta activation as compared to omeprazole alone treated rats. As expected, this reduction in TGF-beta activation is accompanied by a strong reduction of omeprazole-induced Smad-2,3 phosphorylation in the presence of TGF-beta inhibitor, indicating that TGF-beta activation is critical for Smad phosphorylation induced by omeprazole. Also, omeprazole administration significantly induced CTGF and TIMP-1 expression in both mRNA level, figure B, and protein level, figure A. But the most interestingly, that CTGF and TIMP-1 expression induced by omeprazole was significantly attenuated in the presence of TGF-beta inhibitor in both mRNA and protein levels, indicating that omeprazole induced CTGF and TIMP-1 expression in TGF-beta dependent manner. Previously, it has been reported that ROS play an important role in TGF-beta activation and ERK1-2 phosphorylation. So, it was really important to check the effect of omeprazole on oxidative stress markers in rat kidney. And as we can see here, Lipid peroxidation was significantly increased as indicated by an increase in renal MGA content in animals treated with omeprazole as compared to control animals. Furthermore, SOD activity was significantly reduced in animals treated with omeprazole compared with control animals, indicating that omeprazole induced oxidative stress in rat kidney. Coming to the ERK1/2 signaling pathway, omeprazole administration induced a rapid and significant increase in ERK1/2 phosphorylation after 4 hours, which declined rapidly as you can see here. Declined and back to the basal level after 8 hours, indicating that omeprazole activates ERK1/2 signaling cascade in rat kidney with peak at 4 hours. As we can see here, omeprazole administration significantly induced ADAM17 activity after 4 hours as compared to control group, indicating that omeprazole administration induced metalloproteinase activity. In this figure, we found that the administration of omeprazole significantly induced phosphorylation of EGF receptor. On the other hand, the pharmacological inhibitor of EGF receptor caused an almost complete reduction of EGF receptor phosphorylation induced by omeprazole. In addition, ERK1/2 phosphorylation induced by omeprazole was significantly attenuated in the presence of EGF receptor inhibitor, indicating that EGF receptor is critical for ERK1/2 activation induced by omeprazole in rat kidney. As shown here, omeprazole has the ability to induce the expression of cyclin B1. However, this expression of cyclin B1 was highly reduced in the presence of EGF receptor inhibitor, indicating that cell proliferation gene induced by omeprazole depends on EGF receptor tyrosine kinase activity. In conclusion, our findings demonstrate for the first time that omeprazole administration activates not only TGF-beta Smad signaling and subsequent expression of PROFIBROTIC genes CTGF and TIMP-1, but also ERK1/2 signaling cascade and subsequent expression of cell proliferation gene cyclin B1 in rat kidney. Furthermore, this study presents mechanistic relevance of this signaling cascade involving process as indicated by an increase in lipid peroxidation and clear reduction in SOD activity by omeprazole. Finally, this activation of profibrotic and mitogenic cell responses by omeprazole in rat kidney confirm the unwanted effects triggered by proton pump inhibitors. Thank you for listening.